Hello, my friends. This is a quick revision on the practical biochemistry course. To start with, we will give some hints on lab safety. First, we talk about general appearance and attitude. Let's give a reminder on the main points in general appearance and attitude. We have said that we should wear the white coat during the laboratory session. We should also wear skull cap, goggles, gloves, and leather shoes. And all of these preparation or the white coat, the goggles, the gloves, as a sort of protection and some kind of uniform to display integrity inside the laboratory. Also, we give a hint during the main session on no side talks, no eating, no sitting on benches. All our luggages and bags should be kept in lockers. Also to listen, and this is an important note here, and you should keep in mind because it's common in the exam. The correct answer will be always to listen to the instructor or to ask the instructor if you are in doubt doing something, if you are in doubt in the during doing an experiment. For example, you are in doubt that you should heat some chemical or not, so you should ask the instructor if you have to heat such chemicals, or you should you should keep your mix till the color changes appear. For the general work procedure, we said that there are a lot of experimental work and a lot of chemical reactions that could be held in the laboratory. But to simplify, and this is a good word, to simplify all the experimental action or all the experiments inside the laboratory to simple repetitive measures or simple repetitive steps, common steps in or among all chemical reactions. We say that there are four common steps. One of them is to withdraw fluids with pipettes. I don't know, but this is common in Egypt during high school. They used to withdraw a liquid from a test tube using a pipette. But on the other side of the pipette, there's here the mouth of the student. Okay, the mouth of the student. He used to, or students in Egypt used to withdraw liquid from test tubes using the negative pressure of their mouth. But what's correct? The correct thing is to use what's called a glass pipette. Okay, this is what's common or what's correct, to use glass pipette to withdraw fluid. To use dropper, okay, to use dropper, Pasteur dropper or Pasteur pipette, okay. For a small amount or for small volume, we can use automatic pipette. Automatic pipette for for small volume. Okay. You know, a dozen of options rather than to use your mouth to for suction of fluids. Another point, another important point that that occur when we are about to do chemical reaction that includes acid base reaction. Okay, acid base reaction. In such reactions, we need to dilute the acid, okay, or dilute the base, the alkali. So I will use water for such a process, for dilution. So the question will be either you put water first, then the base is added to water, or the opposite, to add acid or alkali first, then water is added after. Okay, so if this comes in the exam in MCQ question, then the answer will be into water first, then acid or alkali is added into water. The third point here is heating is a common step in almost or in many chemical reactions. Heating is used to, you know, increase the energy for particles to clash together. So how can we heat a chemical or liquid? Simply by adding them in, you know, it's a test tube, okay? Adding them in a test tube, and we use what's called Bunsen flame to cause heating or to cause temperature to rise up, okay? Using here a test tube holder, okay, to you know, to carry the test tube in order for the test tube not to drop, okay. So the question will be, where should I direct the mouth of the test tube, or where should I during heating put the test tube toward myself? So if the heating causes the liquid to boil and to evaporate and cause gases and fumes, the fumes will be poured on me. Of course, this is not the correct answer. Should I direct the mouth of the test tube toward my colleague beside me? Again, I don't think it is the correct answer. So the correct answer would be to direct the test tube to space, okay, to space. Another common thing in laboratory, I want to identify, okay, I want to identify some chemical. So such a chemical can be identified maybe by color, right? By color, by you know, by its appearance, okay? By odor, right? There are many chemicals that have odor, even invasive or even hideous odor that can be smelled, or that can be released from such chemical in high concentration, okay, in high concentration. So if I get closer to the bottle inside which the chemical is here, okay, and put my nose here to smell such a chemical. So the high concentration of the chemical will or may cause somehow, irritation in my nose. Okay, can cause, chemical burn, chemical chemical burn, and such chemical burn can diffuse through my nose, through my pharynx, my upper respiratory tract, my maybe my esophagus, can expand to bronchi, So such irritation and the chemical burn that can occur if I put my nose directly to the mouth of the bottle, it can be avoided by what's called waving technique. So what is waving technique? Waving technique is to pull my head away from the bottle and use my hand to cause some waving movement to direct the flow of odor, or to direct the gases toward my nose in low concentration. So I achieve my goal to identify the chemical, and I preclude the chemical burn by narrowing down, or by decreasing the concentration of the chemical odor or the chemical gases that reach my nose. So there are the general work procedure main points. Suction of fluids with pipettes. Pour into water is the right answer. Direct test tube to space, and to smell a chemical by what's called waving technique. Some emergency tips that happen frequently, not only in laboratories, but maybe in while you're walking down streets, or strolling with your friends. Such scenarios, or what we give it importance in our section, the foreign body inoculation, either inside my eye, or inside or the chemical that cause chemical burn. Okay, so the foreign body that is inoculated inside my eye, or the chemical that is inoculated inside my eye, or a chemical that's poured on my skin to cause a skin burn. Okay. Another scenario, when I watch a fainted person in the laboratory, what should I do? So the golden word here is water. So if there's a foreign body that is inoculated inside my eye, or a chemical that is spilled inside my eye, or a chemical that is that's spilled or poured on my skin, causing chemical burn, the correct answer will be to use running water to wash the foreign body, or to dilute the chemical, not to cause more damage. Okay. For the fainted person, like that, there's or the question is, what causes this fainted person to be fainted? And the answer is that there is accumulation of blood in what is called splanchnic area. Okay, what's called splanchnic area. Okay, so when the person is fainted, it's somehow a natural response, so that the blood is flowed back to the head, inside which there is the brain. Okay, there is the brain, the source of consciousness. So if you are in hurry and try to make this person stand up like this, okay, so the blood will continue to flow away from the head, okay, and you will cause more damage, or you will cause the person to feel fainting or to feel more drowsy. So the correct answer will be to elevate the level of legs, to keep this person like this, to keep this person in supine position, and to elevate the legs above the level of the body in order to direct the blood flow toward the brain, okay? If this comes in the MCQ question, the correct answer will be to elevate the legs above the head level. So that was, lab safety in brief, and the points that can come in the exam in MCQ questions. Glasswares, what you should do in glasswares is to revise the table in your practical book. You should identify every glassware, okay, glassware, and you should mention one or two causes for each glassware, okay. So this is a new revise your practical book. For the concentration section, we mentioned, or we discussed percentages, how to represent or to express the concentration of substances dissolved in other. So we will name the substance to be dissolved solute, okay, and the liquid or the other substance that will use to dissolve the solute will be named after solvent, okay. So we have solute and we have solvent. Let's say if the solvent is liquid, like water, okay, and the solvent and the solute, and the solute is particles of NaCl, okay. So we have a solute, solid substance in NaCl that will be dissolved in liquid water. So the solute mass, okay, something solid, will be expressed in grams or in milligrams, okay, and the solvent will be expressed in volume unit, milliliter or liter, okay. So if you want to express the amount of NaCl that is dissolved in a liquid, you will express it like milligrams, okay, in liter, or milligrams in milliliter, okay. Another example, but this time the solute is liquid, okay, and the solvent is liquid. So the two parts here of the solution, the two parts are liquids, like what's called alcohol 70, okay, alcohol 70. What do we mean by alcohol 70? We mean by alcohol 70 milliliter of alcohol that is dissolved in a total volume of 100 milliliter. So if he is asking you how much water in the solution, you will say 30 milliliter. It's a common mistake. that you think 70% alcohol contains 70 milliliter to 100 milliliter water. It's a common mistake, but this is not correct. But what's correct? What's correct that I've just said. We have 70 milliliter of alcohol, added to which a third or 30 milliliter of water to complete 100 milliliter of the whole solution. Then how to express it in the context of percentages? The liquid is expressed as volume unit, so we have either milliliter, we have either liter, okay, and the other liquid again or the same in volume units, milliliter and liter, okay. So milliliter, if I said 7 as I said 7 milliliter of alcohol dissolved in 100 milliliter of the whole solution, then milliliter is deleted or is gone with the other milliliter, and the percent or the result will be 70 percent, like I've said just a couple of minutes ago about the alcohol 70%. Okay, so if a solid is dissolved in a liquid, milligram per liter or milligram per deciliter, a liquid is dissolved in liquid, so the percent here will be the correct expression. For molarity, we have two rules, two important formulas. What is mole? What is mole first? What is mole? Mole is the molecular weight of a substance in grams dissolved in 1 liter. Okay, so this is what we call it mole. Okay, the other important equation is molarity. What is molarity? Number of moles. Okay, number of moles dissolved in the unit or the volume unit. I mean here liter or milliliter or so. How to calculate 1 mole of, or how to prepare 1 mole of substance? By simply knowing the molecular weight of such substance. What is the molecular weight? Mendeleev table that expresses the molecular weight of each substance. Let's say if we have NaCl, Na11, 23, and chloride, from what I remember, here we have 35.5. Okay, so the molecular weight will be the summation of the molecular weight of the substances that share together in that compound. So if we're talking about such a compound, we have 58.5 gram. Okay, like here, like I said, molecular weight in gram, that should be dissolved in liter. So I will prepare 1 liter of water, okay, and I will dissolve 58.5 grams in this liter, so I get 1 mole of NaCl. Okay, and the same if I'm telling you prepare 1 mole of NaOH. Again, you will calculate the molecular weight of each substance sharing their atoms here in the compound, and you will prepare, or you will calculate it in grams, okay, and you will prepare 1 liter, 1 liter of water. You will dissolve such grams in the liter, okay. The other thing is molarity, number of moles. So this is the standard here. This is the standard, and I mean by standard 1 mole, okay. So if I dissolve more than, let's say here in this example in NaCl, I dissolve more grams than 58.5 grams, okay, or less. So the question will be how many moles in such solution. So simply number of moles will be the molecular weight, okay, in the grams in dissolved in the solution divided by the molecular weight per grams for 1 mole, okay. By such calculation, I will get number of moles in the solution, okay. So let's practice on the last two equations. Calculate the molarity of a solution of NaH2PO4, molecular weight. This will come in the exam. He will tell you the molecular weight of the compound. Prepared by dissolving 180 gram NaH2PO4 in 1 liter water. Okay, so molecular weight, you will answer this question like this: equal 180 grams. Okay, he will dissolve grams dissolved equal 180 grams. Volume of solvent equals 1 liter. Okay, so 100, or I will answer it by two methods. Okay, the first method I will say I dissolve 180, okay, in 1 liter, okay, and 1 mole equals 120 in 1 liter, okay. So if I'm telling you how many moles here, this is the definition of molarity, how many moles, then you will divide 180 over 120. You will get, I think, 1.5 moles. Okay. The other question, you will say molarity equals grams dissolved here, okay, on or over the molecular weight per grams for one mole. Okay, so we will complete it like this: gram dissolved 180 over 120. You will get the same result, 1.5 moles. Okay, so this is the first application. Second, if he's asking you how many grams of NaCl are required to make 1 liter of 0.05 mole. Okay, so I want to prepare 0.05 mole equals grams dissolved over the molecular weight in grams for 1 mole. Okay, so 0.05 here express the molarity. Okay, you will continue like this: 0.05 mole grams dissolved. This is the the X, or this is the aim of the question. Molecular weight in grams, we have 58.5. You will do the math, multiply this value by 0.05, and you will get the grams to be dissolved. Okay, how many grams of NaCl are required to make 100 or 1500 milliliter of 2 mole NaCl? So you will first define or detect or identify molarity. Molarity will be 2 mole. Molecular weight grams equal 58.5 grams. Okay, volume of the solvent of the solvent equal 1.5 liter. Okay, see what I'm doing here. I converted the 1500 milliliter to 1.5 liter. Okay, so molarity equal gram dissolved on molecular weight in grams. Okay, molarity here is 2 mole. Gram dissolved, this is what we are calculating. Molecular weight in grams equal 58.5. So the gram dissolved in 1 over 58.5 will be, I guess, 117 grams. Okay, but this is for 1 liter. Okay, so if you want it for 1.5 liter, you will multiply 117 by 1.5, and you will get your math and reach your results. Okay, another part of concentration is called normality. What do we mean by normality? Why we need normality? We need normality in acid, or mainly in acid-base reactions. Let's say we have here 2 hydrogen atom like H2SO4, okay, and we have here NaOH. So like this. So this is OH, this is OH. In acid-base reaction, I want the number of H to roughly equal number of OH, so that the chemical reaction will be directed to stability, or to the same, or with no changes in acidity or alkalinity. So if I want to prepare, and let's concentrate or keep in mind this point. If I want to prepare gram equivalent per weight, and this means to prepare grams that can react with the other side of OH. So I need to divide moles, or the grams in mole, over the valency. What does we mean? What do we mean by valency? The valency is the number of hydrogen here, okay, number of protons. So I want to divide the 2H over 2, okay, the valency will be 2, right? So I get 1H that can react with the 1OH here, okay? This is the aim of normality, to normalize the number of hydrogen with the number of OH, okay? To put this in terms, to divide the gram in moles over the valency, okay? And this is the equation number 1. But there are other scenarios. The question is how much this acid or alkali, or alkali is powerful, okay? This is the question. How much is this acid or alkali powerful? Okay? What does it mean? It means that how many H or how many OH this acid or alkali gives out to the reaction. Regarding our example, we have H2SO4, so we have 2H, okay? So the normality will be 2. This is, as simple as it is. But to put this in terms, so normality equals molarity multiplied by valency. Okay? So to apply this in our example, H2SO4, so you will multiply the molarity by 2 and get the normality. Okay? Calculate the molarity of 0.02 normal H2SO4. To answer this question, you should write this formula. This question is about how much powerful is this solution. Okay? So the equation will be the second. The equation used will be the second one. Normality, okay, equals molarity multiplied by valency. Okay? Valency here will be got from the chemical formula of the acid. We have H2, we have 2H. Okay, so the valency will be 2 multiplied by molarity. This is what I want to get. So this will be written down like this, and the normality is 0.02 normal. So molarity equals 0.02 over 2. So the answer will be 0.01 mole. Okay, and this is the correct answer. Dilution, another concept in concentration. Dilution, two major parts you should understand in dilution. Number one, what is the dilution factor? What is the dilution factor? Okay, there's a common mistake that actually or that commonly occur. What is it? You calculate the dilution factors as ratios between the initial volume and the volume added. Okay? This is not correct. Let's give an example. If I want to dilute in, in such a cylinder, if I put here 10 milliliter of a concentrated solution, okay, and I want to dilute this solution by distilled water, okay, distilled water up to 100 milliliter. So what am I doing here? I want to the particles that are confined to, or that are located in 10 milliliter to diffuse, okay, to diffuse in a wider space up to 100 milliliter, so that I get what's called dilution. I want to dilute the sample here 10 milliliter, okay? So the dilution factor will be calculated as the final volume over the initial volume. So the final volume here is 100 milliliter over 10 milliliter equal 10 times. So this is the dilution factor. I dilute the sample 10 times its concentration. Okay? The common mistake, as I said, is to calculate the dilution factor as a ratio between the distilled water and the volume of the sample. This is not correct. This is not correct. So if you answer it like I am adding 90 milliliter to 10 milliliter, so the dilution is 9. So this is not correct answer. The correct answer is to calculate the final volume 100 milliliter and to divide it over the initial volume. A quick reminder to what we have discussed on alcohol 70. Remember, we said the same. We give a notice on that the 70 milliliter of alcohol is dissolved in a total volume of 100 milliliter, not a total volume of 70, 100 or 70 plus 100 milliliter water. So the total will be 170 milliliter. This is not correct, right? So the 70 milliliter is added to 30 milliliter water to reach a total volume of 100 milliliter. It is the same concept like when we are discussing the dilution factor, to divide the final volume over the initial volume, not to use ratios. Okay? After dilution factor, this is the principle number 1 or the note number 1 to study. The note number 2 is to remember this equation. C1V1 equals C2V2. Like I said here, if I want to dilute a sample, so I will use, instead of C1I for initial, VI for initial. So this is the initial concentration and this is the initial volume, equals C2, the final, the final, for the final here, C final and V final. Okay, it's a common sense that the final concentration will be less than the initial concentration because I want to dilute a sample. Okay, let's give an example for this. Uh, if 15 milliliter of blood, so I will use my imagination. This is 15 milliliter of blood, okay? And I want to complete this volume to 100 milliliter, 150 milliliter, like this. So we have a final volume 150 milliliter and we have initial volume 15. So the answer of the dilution factor is final volume over initial volume 150 over 15 equal 10. So I dilute the blood 10 times. If I want to prepare 100 milliliter of 0.25 mole of NaCl from a stock 0.5 mole. So what I'm not seeing here is the stock concentration 0.05 and the concentration I want to reach is 0.25. So this concentration is less than the concentration 0.5. I want to dilute 0.5 to reach 0.25. Okay? The question is about the initial volume. Okay? So if I apply to the law or the formula CI equals C final V final. CI will be 0.5 mole, okay? VI, this is the volume I want to withdraw or I want to get out from the stock. CF will be 0.25 multiplied by 100 milliliter, the final volume. So VI equals 0.25 over 0.5 multiplied by 100 milliliter. The answer will be half multiplied by 100 milliliter, so I get 50 milliliter. If I stopped at this point, I will get half the marks of the question. So what should I do? Let's use our imagination again. Now you move or you relocate 50 milliliter of the stock 0.5 mole. So you will get the initial volume. But after this, to dilute this 50 milliliter in what he is asking you to do 100 milliliter, so you will add distilled water here, okay? So you can get the final volume of 100 milliliter, okay? In only this way, you dissolve it or you make this particle diffuse into space to be diluted. So another line will be added here. You will say, after you get an initial volume 50 milliliter, distilled water is added to reach a total volume, a total final volume of 100 milliliter. Okay? So the law, the formula, how to apply to the law, how you get your calculus and distilled water is added to reach the final volume. Section number 3 was about solutions. Okay. The most important feature in solution is to differentiate and discriminate between the different kinds of solution. So if there are particles less than 1 nanometer, like those particles, okay? So the size of such particles will allow them to completely dissolve it in the solution, and you will get a clear aspect. You will get a clear aspect. It's called also true solution. Okay, this is true solution. You will get a complete dissolution of the particles inside the solvent. So we call this type of solution crystalloid. Okay, and this is number 1. Number 2, if the size gets larger than 1, up to 200 nanometer, this size will allow the particles to be partially dissolved in the solvent. Okay, so you will get floating particles here. Okay, not completely dissolved and not to sediment. Okay, just floating. In this scenario, or in this case, you will get what's called colloid solution. If the size gets larger than 200 nanometer, then the particles that size will not be allowed to dissolve, neither completely, neither partially, and will sediment eventually in the or at the bottom of the test tube, what's called number 3 suspension. Okay, at this point it's good, but if you asked about examples for each one, so for crystalloids, we'll mention NaCl. It's like, you know, the food salts, right? NaCl. NaCl is completely dissolved. You can also mention glucose. If you get glucose into water, it's completely dissolved. Okay. Colloid, you can mention in colloid starch, and it's frequently to see starch in the exam, or albumin protein. Okay. For suspension, you can mention sands. Okay, so the size of particles, the type of solution, and 1 or 2 examples for each one. Dialysing bag. What is dialysing bag? Dialysing bag is a bag, in essence. Okay, this bag is loaded with water, at which there is particles of crystalloid and particles of colloid added to water, and all of this inside the bag. Okay, this bag is like the dialysing membrane in our kidneys. Okay, dialysing membrane in our kidneys. This dialysis membrane is or has to filtrate the blood, okay, get the wastes inside the tubules to be washed in urine, and to keep albumin inside the blood, okay. So the dialysis bag, the coat or the cover or the outlining of the dialysis bag is manufactured and synthesized the same way the true dialysis membrane. So if there is a normal dialysis bag, what are the outcomes that you are expecting to see? The crystalloid will pass through the dialysis bag because their size is low, okay, and the colloid, because their size is large, will be kept inside the bag, right? So if I put this bag in a beaker this way and notice the diffusible water into this space of the beaker, what I'm expecting that here I will get crystalloid. Crystalloids will easily dissolve through the bag to be here in the space between the bag and the beaker, okay? But for colloids will be kept inside the bag. Yeah. So if I am simulating here, colloids will be kept inside the bag because their sizes are large. Back again. So keep in mind that crystalloids, maybe NaCl, maybe glucose, will be out of the dialysis bag, but colloids like albumin, like starch, will be kept inside the bag in case that the dialysis bag is working and manufactured properly. Biomolecules test. This is the slide is continual, or it's it's a further step, a next step for what we are expecting, or a further step to test the components of the diffused solvent or of the diffused water here, okay? So if I want to test glucose, okay, the glucose that is supposed to diffuse here with crystalloid or as a crystalloid, and protein that is normally kept here, and the starch, the same as protein, colloid. So I want to test each substance, okay? So for glucose, there's what's called Benedict test. Benedict test, the Benedict reagent contains cupric sulfate, CuSO4. Being added to glucose is having a reducing group. This reducing group will reduce copper sulfate into cuprous oxide, will reduce the Cu plus 2 or 2 plus to Cu plus 1 plus. Cu1 plus added to oxygen will cause cupric oxide, like this, okay? The copper oxide will precipitate, as you see in the picture. If there's a small amount of glucose, so the precipitate will be greenish. If there's a moderate amount, the precipitate will be yellowish. If there's a large amount of glucose, the precipitate will be from orange to red. Okay? To get the precipitate, you must break down the bonds between Cu and SO4, and to allow for the glucose to reduce cupric into cuprous, so you need to cause heating. Okay, heating in the exam will be at the bottom of the test tube. Okay, you will heat at the bottom of the test tube. Okay, so how to do it in the exam? You know now that Benedict is for glucose. You know the principle. You know that you will heat. Okay, you know somehow the observation. Okay, but how will you do it? You do it like this. You will put the reagent first. If there's a test tube, you will put, you know, fourth of the test tube. Okay, it is like 5 milliliter. Okay, fourth the test tube, and you will add 8 drops of the sample. You will add 8 drops of sample to 5 milliliter of the reagent. Okay, and this mix will be heated from the bottom of the test tube till you notice the mix is boiling. Okay, the mix is boiling. If after boiling the precipitate appear, yes, it's glucose. There was glucose. But after boiling, the precipitate doesn't appear. So this in this case, the conclusion will be negative for glucose. Okay. If you are asked about the principle, you will mention it. If you are not asked, don't mention it. Just put your measurement. Okay. Sample 0.5 or 8 drops, reagent 5 milliliter, observation and result. The observation differs from result. How is that? Observation is simply what you observe. Okay, so if I'm asking you what do you observe here, precipitate, greenish or yellowish or reddish according to the amount of glucose. But if you are asked about the result, you will say, yes, I conclude, I deduce from this observation that there is glucose. So your observation will be precipitate, but the result will be either positive for glucose, or if precipitates appear, or negative for glucose if the precipitate doesn't appear after boiling of the mix. Okay, so observation is what you notice, but results are what you conclude or what you deduce. Okay, Pirate, we say it like this. We say like pirate for protein that will cause the color change to violet. So pirate, protein, violet, and 8 drops. You notice that the letter T is frequent in such test. Pirate, violet, protein, 8 drops. So the substance with pirate test is protein. Pirate is used for detection or for identification of protein in the sample. Pirate reacts with the peptide bonds, so it's specific for protein which has peptide bonds. No other biological compounds that contain peptide bonds rather than protein, so it's specific for protein. Sample will be 2 milliliter like this, and you will add using dropper 8 drops. There are 9. I will delete 1. 1, 2, 3, 4, 5, 6, 7, 8. 8 drops to the sample. No need for heating. The color will be changed immediately. So again, you will write down your measurements. 2 milliliter added to which there are 8 drops. Observation color changes, not precipitate. Precipitate is with Benedict, but color changes to violet with Pirate. If there is color change to violet, you will say positive for Pirate, like in this example or this picture. But if the color stay bluish, you will say negative for Pirate. Okay. For iodine, it's like the potato experiment in our preliminary stage or in our primary school. If there's a potato and you put on the potato drops of iodine, the potato will get blackish, right? So it's the same. Iodine is used to detect or to identify starch. Potassium iodine reacts with the starch. The mixture will be like this: 2 milliliter of the sample, okay, added to which 2 or 3 drops of the iodine, okay. If the color it changes to purple black, like this one, this is purple black, okay. If the color changes to purple black, so the result will be positive for iodine, positive for starch, I'm sorry, this is a starch, positive for starch, okay. And if the color, this is protein, the same, okay, the same mistake, positive for starch. And if there's no color changes, so your result will be negative for starch, okay. Just memorize the measurement, your observation, and keep in mind that observation is different from conclusion. Observation, what you see, what you notice, but conclusion, what you deduce from your observation, okay. You will express it like this. Benedict, you should mention there's precipitate. The other 2 tests, color changes. If the color changes occur, positive for for substance. If there's no color changes, then negative for the substance, either protein or either starch, okay. In some scenarios in the exam, he will put a dialyzing bag like this and ask you to test for protein, glucose, and starch in the filtered. Here in this filtered, you will be asked to test for the three substances in the filtered. So what do you expect, or what will you do? You will do Pirate, okay, the same way we have mentioned. You will do Benedict. You will do iodine test, okay. So you expect, if this dialyzing bag is working properly, it will allow glucose to diffuse, but will keep protein and starch inside. So you expect that Pirate test will turn negative, okay, no color changes. And Benedict, there will be precipitate, so it will be positive for glucose. Iodine will turn negative and no color changes. Okay. So if you ask it in this section like this, you will do the three tests or whatever the test he's asking you, and you will write down your observation and your conclusion. Okay. The last part is spectrophotometer. Okay. Simply, if you have a cup of tea and this cup of tea here is exposed to light, the darker the tea, okay, the tea, you expect that is more concentrated, right? And the lighter, less concentrated. This is the simple rule for spectrophotometer. The absorbance that is responsible for either the tea or the solution you are measuring or you are estimating its concentration. If the absorbance of light increases, that causes the solution to get darker. If the absorbance of light decreased, okay, then or decreases, then it will cause the lighter appearance of the solution. Okay. If I'm asked to, to express what I'm seeing as a human being, I will say this solution is darker than this one. Okay, this solution is darker, but this solution is lighter. Okay, this is what the gross eye of the human being is allowed to say, but my eyes are not allowed to say that this solution is 5 gram per deciliter, right? There's 5 gram per deciliter tea. Okay, so this is not or this is beyond my abilities as a human being. So we should use a device. This device using photosensitive parts that will detect the light sensitively, okay, light sensitively, and will record the result in digital meter, okay, in digital meter, so that I can get the concentration precisely. This is the major or the main aspect. So keep in mind that absorbance of light that is responsible for the density or intensity of the light is directly proportionate to the concentration of the substance inside the solution, is directly proportionate to the concentration. Okay. From the name spectrophotometer, we can say that photon to be metered, to be measured, is to measure what we just explained. The color it changes, okay, either darker or lighter. Okay, so this is the meaning of photometer. But what does it mean to say spectrometer? We say that we should make the incident light, simply, without any details, the incident light that is allowed to be incident on such a solution should be determined or should be maintained or selected at wavelength, okay, this wavelength is complementary to the color of solution. Why? Why? Because at this length it will be maximal absorbance of the color. Okay? So spectrophotometer, photo, the color changes. Spectro means that I will choose from the light spectrum, a specific wavelength at which there is maximal absorption of the solution. Okay? So let's revise simply the spectrophotometer parts. We need, in my example, a cup of tea. Instead of the cup of tea, I will put the solution inside test tube. Instead of my eyes, I will use photosensitive cell. Okay, photosensitive cell. And this photosensitive cell is connected to a digital meter. Okay, so this is meter. Instead of the light coming from the sun, I will use light source, tungsten lamp, and a wavelength selector to select a specific wavelength from the spectrum, from the light spectrum. And here I will use a slit. It is something like the slit of the camera to control the intensity of light. Okay, that is allowed to be incident on the test tube. Here in this part, the solution will cause absorbance. Okay, and the transmitted light will reach the photosensitive cells. So by diminishing the transmitted light from the incident light, I will get the absorbance that's caused by the particles of the solution, and by the equation that absorbance is directly proportional to the concentration, it will be allowed to me to reach or to calculate the concentration of the substance. Okay, but there's problems, or some problems on estimating serum or plasma analyte. Problem number one is these analytes means the thing or the substance to be measured, or I want to know its concentration, okay, inside my body, dissolved in serum or in plasma. These analytes are colorless. Okay, is this a problem? Yes. Why? Because the spectrophotometer depends on the absorbance of color and color changes. Okay, so for this analyze to be colorless, I have to put another reagent. This reagent may be something to bind with the analyte, or a substrate at which the analyte will work to produce products, to produce colorful products. Okay, the net result is adding reagent to the colorless analyte will turn the colorless analyte into colorful analytes. Okay, so in the case of protein, if I add to protein violet, then I get violet color, right? So the violet color here is something colorful. I notice it's a color, but protein is colorless. Okay, this is an example for what I want to say. But here there's another problem. If I put the protein sample added to the Biorite reagent, if I put it to the device here, the spectrophotometer, the digital meter will calculate the absorbance by what? The sample, the protein, and the Biorite. The question is, do I want to get the absorbance and the concentration of the sample, or do I want to get the absorbance or the concentration of the sample added to reagent? Actually, I want to measure the concentration of the sample only, because this is what matters. Okay, this is what I want to measure, this analyte concentration in, right? So I want to cancel the interference that is done by Biorite. How can I do this? Simply by putting the Biorite reagent first, okay? Then I get its absorbance and I get its concentration, I get the interference that is caused by Biorite. After this is the step number 1. After this, I will put Biorite added to sample. So the reading here is for Biorite only, but the reading here is for Biorite plus sample. Simply by diminishing the absorbance here from the total sample plus Biorite, I will get the concentration of sample only, okay, or the absorbance of sample only. This process is called to set the device to 0, which means I put the Biorite reagent first into the device and diminish this value from the device to make it 0, so that if I use Biorite plus sample, I will get the reading that's caused by the sample. Okay? So to work or to measure an analyte, let's say for example protein, I will need to prepare 3 test tubes, okay, like this. The test tube number 1, I will name it blank. Test tube number 2, I will name it standard. Test tube number 3, I will name it the sample I want to measure the concentration of analyte in. After this, the blank will be used to setting the device to 0, like we have just explained. So I need to put inside the blank reagent only, but added to it distilled water. Okay, distilled water, it has no particles to interfere with the light, but the reagent will be only the responsible for the interference inside the spectrophotometer. So we know the use of blank. Okay, the sample will be prepared by putting an equal amount of the reagent added to the sample. Okay, so this is the sample. The standard will be like this: a standard plus reagent. I know the use of the blank to set the device to 0, okay? The use of the sample to measure the absorbance and then the concentration of the analyte inside the sample. But what is the use of the standard? Two uses. Use number one is the standard. I prepared the standard already, okay? I am the one who prepared the standard. So I know the concentration of the standard. I know it well. And I know the absorption of the standard. I don't need the device to calculate it. So why is it used? It's like reversing the procedure. It is like to put the standard into the device. So if the device read the concentration, then I check that the device is working properly. But if the device reads the reading or the measurement that is expected from the standard wrongly, then first I should fix this device before using it for reading my sample. So the first use is to check that the device is working properly by comparing the reading of the device with the actual reading of the standard I already prepared. The second use is by knowing the concentration and the absorbance. Remember when we said that absorbance is directly proportionate to concentration. Okay? So if I have a standard, I know its absorbance. This is the standard, okay? I know its absorbance that lead to concentration of a standard, okay? So if I want to know the absorbance of test, that will lead to concentration of test. By using simple math, I will do, cross matching, okay? So absorbance of standard will be multiplied by the concentration of test, and the result will be divided over concentration of the standard to get absorbance. And the same way, if I want to get concentration of test, I will multiply concentration of standard by absorbance of test and divide the result on absorbance of test. Okay? So again, the blank is used for setting the device to 0. The standard is used for checking the device is working properly, and its measures will be used in the cross matching in the math needed to calculate the concentration of the test. Okay? So again, how to calculate the concentration? By the equation we just deduced, concentration of test equals absorbance of test over absorbance of standard multiplied by the concentration of standard. Okay? The value absorbance of standard, I know it because I'm the one who prepared it. The concentration of standard the same. The absorbance of the test, I will get it from the spectrophotometer. Okay? And the concentration of the test will be reached by applying this law to the case scenario. Okay? Uh, if you asked in the exam about to estimate the total protein, okay, then you will apply the same steps but on protein. Here the test tubes, the reagent will be pyrite, okay, and the same for other things. Distilled water will be distilled water, standard, sample, the sample you want to measure your protein in. So the only change is will be the pyrite will be the reagent, or will be the used reagent. Okay. By the same steps, you will get the concentration. You will get the concentration of the sample. You will get a value. The value unit, keep in mind, it is in gram per deciliter. Protein is a molecule of large size. And you will compare your value, your reached value, with the normal range of protein. The normal range from 6 to 8 gram per deciliter. If the value is below 6, then it is below 6. It is hypoproteinemia. If the value above 8, it is hyperproteinemia. The classic causes of hyperproteinemia: malnutrition, okay, or there's good nutrition but there's malabsorption. Mal means poor absorption, inefficient absorption, or there's good nutrition and good absorption, but the loss of protein is increased in nephrotic syndrome. Nephrotic means, or is relevant to nephrons, okay, nephrons relevant to the kidney, okay. So nephrotic syndrome, or there is impaired senses in chronic liver disease. The hepatocyte is no longer able to produce albumin, okay. So hypoproteinemia, malnutrition, malabsorption, nephrotic relevant to nephrons, or impaired senses in chronic liver failure or chronic liver disease. Hyperproteinemia, the reasons behind hyperproteinemia lie mostly in immunoglobulins. Immunoglobulins, as the name denotes, is something relevant to immune system. The thing that is responsible for the immune response or the defense action against invading pathogens. So I am expecting that immunoglobulins will rise up in cases of infections, in cases of invading pathogens, invading microbes inside my body, or in chronic inflammation to supply the blood with immunoglobulins, okay? So this is non-specific elevation of immunoglobulins. But if there's a certain immunoglobulins, IgM, immunoglobulin M, okay? IgM is elevated with cases of something that starts with M, multiple myeloma, okay? Multiple myeloma is something, or is a type of, malignant case, or a tumor case. There is a tumor, or there is overproduction of immunoglobulin M from my bone marrow, okay? Bone marrow, it is inside my bones, the thing that is responsible for creating the immune cells, okay? So bone marrow will be exaggerated in multiple myeloma in the context of overproduction of immunoglobulin IgM. Okay? Dehydration can be a cause for hyperproteinemia, but it is not clinically significant. If I am imagining that is plasma, that plasma is the solvent and the albumin is the solute, okay? So if the amount of water or decreased in plasma, this will cause the solute to relatively increase in concentration. So it's relatively increase in concentration of protein in cases of dehydration at which there is a decline in water content. Okay, and again it is of minimal clinical significance. The last part that you can be asked in the exam to measure a certain volume in a cylinder. Owing to the surface tension, liquid surface mostly is or tend to be curved downward like this, to be curved downward to create a meniscus. Okay, so if you are asked to measure this volume, will you read the graduation at the raised edges at the periphery, or will you read the graduation at the lowest point? The correct answer is the lowest point. You will make your eye at the level of the lowest point and record your measurement. Okay? So this is how you get your correct volume. Thank you.